Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Ivana Petrovich Kashmak from BIA Separations, now a Sartorius company.
Hello to everyone, and thank you for introducing me, Leah. Today's topic is going to be the analytical tools to improve empty-full ratio in initial stages of AAV process development. So this is the work done by our upstream process development team as well as the analytics team. A little bit about BIA Separations. So we are the leading developer of monolith technology, and we're the exclusive producer of convective interaction media chromatographic columns. Since a little bit more than a year ago, we are now part of Sartorius family, and we are their center of excellence in gene therapy. Our products are specially used in gene therapy and vaccine markets, and we supply unique monolithic chromatographic columns which are complementary to porous particles and membranes. We are, as you can imagine, the experts in bioproduct manufacturing process development. So we have over 30 cGMP processes that includes plasmid DNA, messenger RNA, also virus downstream processes that we transfer to different CMOs and sponsors. Some projects also include the ones with coronavirus. We have more than 20 years of experience with adenovirus, with almost all serotypes of AAV, with influenza virus, vaccinia, exosomes, bacteriophages, virus-like particles, and many, many more. So what is our technology about? It is a special media which we use to make our chromatographic columns that allow very unique approach. On this slide, you see on the left side the traditional approach with porous particles. Porous particles have many advantages. However, if you would like to increase the speed of your process, you're quite frequently forced to sacrifice the capacity. On the other hand, if you want a better resolution, you might need to have a lot slower process. This can particularly be a problem when you're dealing with the larger biomolecules. On the other hand, monolithic columns come very handy to solve this problem. In monolithic columns, we have channels rather than pores. There are no dead-end pores, and all the channels are connected to each other. All of the surface is easily accessible for large molecules, which allows us to have high capacity. This kind of design avoids the entrapment of solutes, and all of the molecules are passing with the, let's say, without the turbulent flow. We have very good resolution, which is flow independent, which means that you can go as fast as you want. There are, of course, certain limitations when it comes to pressure, but our housing and the monolith itself can withstand quite high pressure. So usually the limiting factor is going to be the sensibility of your biomolecule to the pressure. Our columns come in many different chemistries, some of them being traditional, such as quaternary amines, cation and anion exchangers, but we also have specialized columns for purifying plasmid DNA, adenovirus, adeno-associated virus, and others. Our columns exist in same chemistries, both for purification purposes as well as the analytical purposes, as the ones that you are seeing right now on our slide. These analytical columns are combined with our HPLC system called PATFIX, and we commonly use them for quality control, for plasmid DNA, for transfection experiments in upstream, as well as recording background with media, feed, cells You can also analyze AAV, its expression profile, upstream, downstream samples, and you can also use it for product release tests. Now, the main challenge that we face in bioprocessing nowadays is upstream and downstream being observed as separate processes instead of two parts of the same process. In order to go forward, we do really need to consider the holistic approach. For that purpose, we were in need for fast and reliable analytics, as well as some orthogonal methods which would allow us to work with upstream and downstream samples at the same time in order to develop a high efficient platform for virus product manufacturing. HPLC analytical methods came as a natural answer to this question because they allow us to analyze the product as well as see the distribution of impurities and also access the empty-full capsid ratio. For this particular purpose of testing upstream samples, which are rather complex compared to downstream samples, we developed a novel technique, which is called PATFIX HPLC valve switching. This is not an ordinary HPLC instrument in sense that it contains not one but two columns that are combined together, and there is a valve which is able to switch between them. First column is a cation exchange, which serves to capture the adeno-associated virus, and the second column is the one that actually performs the empty full analytics. What this method allows you is to obtain the relative titer between the two samples, to also obtain the absolute titer, but for this purpose calibration curve is needed, as for qPCR, for example. It allows you to see the empty-full ratio in each sample, and you can also pair it with the additional detectors, such as the tryptophan fluorescence, and you can also stain with picogreen for DNA content. This approach, since it consists of two columns, one of them serving for pre-purification and the other for analytics, it is suitable for all complex samples, not only from upstream, but also from early, midstream, and downstream samples. That includes any kind of spent media, light cell pellet, whole harvest, and so on. So here is how it works. You can see on the left side chromatogram coming from the first cation exchange chromatography. Sample is being bound to the first column and then is being eluted in a step gradient using pH. This allows us to send the sample through the valve to the second column, which is an ion exchange, and bind it to it directly. In the middle, you can see the chromatogram coming from an anion exchange, where we elute our bound pre-purified AAV using the classical salt gradient. The anion exchange fraction zoom in, you can see on the right part of the screen, first peak being the empty and the second one being full particle. For that purpose of analytics, we use UV 260 and 280, as well as a multi-angle light detector, which in this case we do not use it to do additional analytics, but we use it as a detector of the particles. So on the right side of this slide, in the red rectangle, you see a comparison of most signals coming from three different samples. First one of them being our AAV8 standard. So this is a sample that has been captured on a cation exchange, and you can see both empties and fulls. The second one is an optimized harvest of AAV8, where you can see a slightly different empty-full ratio. This one comes from SF9 cells. And the final one on the right side is our AAV8 harvest from HEK cells, which has been in the process of the optimization. It has rather high percent of full particles, as you can see in the second peak, but unfortunately low titer. So this is a work in progress. So on following slides, I'm going to show you several examples of how we use this analytics to do the AAV upstream process optimization for empty-full ratio already at the very low sample size. So these have been performed in a shake flask, but you can also couple it with amber 15 or even amber 250. In the red rectangle, you see multi-angle light scattering of two different samples. The only difference between them is that complexation between plasmid DNA and transfection reagent was made either at the room temperature or 37 degrees. On the right side of this slide, you see a comparison between ddPCR titers and the quantification from moles. Here we did not observe a big difference between the two samples. However, already on the following slide, you see the comparison of the incubation time of a complex between DNA and transfection reagent, one of them being 15 minutes and the other being 30. Here we notice a small difference in the sense that a 15-minute incubation gave slightly lower quantity of the full AAVs, and it gave slightly higher quantity of the empties. Now, here the difference already starts being very clear. In this experiment, we tested one of the transfection reagents that we work with, and we combined it with a different concentration of DNA. So plasmid ratio always remains the same. It's just the concentration which is different. And you can see when we use the smaller concentration of DNA, we get significantly higher percent of full AAV capsids. However, if you would only look at the full peak as well as PCR results, you would see that the smaller quantity of DNA in our case resulted in less total in less full AAVs. However, if we know that empty AAVs represent an impurity that should be removed, one might consider optimizing this a little bit further and maybe rather opting for a case where we have slightly lower titer but significantly less portion of empties. On the following slide, you can see a similar experiment being done with a different transfection reagent, but here different parameters were kept constant. So for this experiment, we kept the viable cell density constant, and in the previous one, we kept DNA per million of viable cells constant. But the trend is observed in a similar way. The less DNA you use, you have less of empty particles. This is not to say that this is how it's going to work in your experiments. It is always a good idea that you test your conditions, particular plasmids that you use, and your transfection reagents, and see if you observe the same. In this experiment, we tested the different ratio between plasmid DNA and transfection reagent, 1:5 being extraordinary high ratio, which is not frequently seen in industrial applications. So in our experiments, we obtained almost identical titers of the full AAV capsids, but when more transfection reagent was used, we observed more of empty AAVs. Finally, we can also use the same analytical approach to test the secretion kinetics during the upstream process. These are results from our adherent upstream process where we collected samples at different times. And you can see as the time progresses, you observe differences in empty full production. The interesting part here is that the empties start being produced first. The final part, which is still under construction, is a technique that allows us to detect other subpopulations of AAVs and not just empty full. So this technique is a combination of the ultracentrifuge with an HPLC system, where after the ultracentrifuge separation of particles, we use the pumps to pass the sample directly through our detectors. On the graph on the right side, you can see first the full particles and then the empties coming. Now, this is a rather complex slide, so I will only go to the top right part of it, which says HPLC zoom in. So on this particular one, you can see that using this method, you can notice not only full and empties, but also partials, which show up as a shoulder to full particles. We think that these could be partially full capsids that can contain various lengths of DNA inside. The further investigation is ongoing. I would like to leave you with the final conclusions of what I showed today in this presentation, which is an analytical tool which allows us holistic approach to vector manufacturing process that combines upstream and downstream in one. This method is orthogonal to ddPCR ELISA approach for empty full, as well as ddPCR for determining the total titer of your AAV sample. It requires rather small sample, which is up to 10 milliliters volume, and this is suitable for testing samples very early in your process development. Finally, this kind of approach allows you to make the optimization early on to reduce empty and partial AAV particles as soon as possible. Thank you for your attention, and I'm happy to take questions.
Okay, great. Thanks, Ivana. So, how much time does it take to analyze one sample?
So the full analytics of a single sample from the moment the sample is prepared takes about 1 hour. This is not to say that you have to be present all of the time during that analytics. It is sufficient that you prepare samples, you leave them on the autosampler tray, and you can control the machine and the analytics also from your computer, from the office, or from home.
Okay, and as a reminder, you can go ahead and type your questions in for Ivana. And if we don't get to your question today, we will be passing those on and she can follow up with you directly. The next question is PAT Fix software available in a demo version?
Yes, definitely. So if you would go for Bioseparation's website, there you would be able to find the PatFix software demo for an online trial. So there we have preloaded samples that you can test and play. Those include plasmid DNA, AAV, as well as mRNA. You are also going to be able to find different tutorials and videos on YouTube dedicated to our newest version of the PatFix software.
Okay, so it looks like we just have one more question. Um, so go ahead and keep typing your questions in. What other analytics do you use for the upstream process development?
So as I mentioned in this talk, it is very important to have more than just one method. So ideal is to have several orthogonal methods and also complementary ones. So we commonly rely when it comes to AAV upstream on ddPCR. We do valve switch for empty full. We also do size exclusion analytics. We measure DNA content with a picogreen staining, usually combined with different chromatographic methods. We make a total AAV quantification using the CIMAC SO3, which is cation exchange analytics, and we also have different plate tests available for total DNA and total protein.
Okay, we have another question. Is it possible to characterize AAV without a Mols detector?
Yes, it is possible. So, as you could see on one of my slides, when the titer of AAV in your sample is high enough, that is, when the quantity of your AAV is higher than the background, you can reliably use UV signal. However, when the titers are lower and our LOD is as low as about uh, let's say about 2E9 of total vector particles per milliliter if you use the maximum volume of the sample. So in that case, we prefer to use moles because of higher sensitivity. Otherwise, if you have good concentration already in your sample, you can definitely use UV 260 and 280. I just have to note that in that case, you need to use a correction factor. Also for moles, you need to use one because the empty and the fulls will have different extinction coefficients.
Okay. Well, thank you, Ivana, and thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.